This is Symposium Session 21, Preclinical Assessments of Novel Genetic Therapies, so hopefully you're in the right space. And just to highlight who we have coming up, the first speaker will be me, Kara Fauché. I am Senior Director of Gene Therapies at the CF Foundation. I'll be followed by Wanda O'Neill, who will talk about small animal models, John Engelhardt, who will talk about large animal models, and then Deepika Polanini, who will talk about early clinical trials. At the end of that, we'll have a Q&A session moderated by JP Clancy, who is also making his way down from the plenary. So I had this housekeeping slide that said, save your questions for the Q&A and put them in the app. We'll see if that works. If not, it's a small enough room, I think you can raise your hand and stand up. And then I was asked to kick this off with a quick slide on why a preclinical assay matrix is important. And I think following the plenary, that's not hard. So we heard Nicole talk about how many trials we have moving forward. And we heard from patients how desperate they are for really good therapies. And so the whole reason we need good clinical trials is for those patients. And how do you have a good clinical trial? Well, you set up an infrastructure to support development of good preclinical packages so that when those drug products get to the clinic, we can be confident that they'll have biological efficacy and hopefully improve clinical outcomes. So I had this slide, which I'm not really talking to because I'm talking about the plenary, but I was going to talk about the history of genetic therapies trials in CF, and there have been a lot. And we were talking the other day about, we should go back and look through all of them and compare why this one showed this and this one showed that. And I think what stood out to me was that they were really varied. They targeted different tissues, nose, sinus, lung. They maybe saw CFTR expression, they maybe didn't. They didn't necessarily show CFTR function, and they used different endpoints. And so it's really hard to compare what might work, what went wrong, if things are done in such a varied fashion. And so that's my plug for why we need to standardize preclinical package development so we have confidence in what's moving forward for those patients. All right, and with that, I will actually go to my real slides. Maybe. Okay. And I apologize for the weird capitalization and font that was not how it looked on my computer. So the topic of the first talk will be model systems and developing an assay matrix or approaches to building a toolbox for preclinical package development. Here is my disclosure slide. I have nothing to disclose. And then the agenda for my talk. So I'll start with what we can learn from the past, not clinical genetic therapy trials, but from modulators. I'll talk about the complexities of genetic therapies for CF, the preclinical data framework that we're developing, the models and tools we have today, and the guidance we've put out currently, and then looking toward the future, how that might change. So what can we learn from other drugs and other diseases? CFTR modulators are an incredible success story. And the cool thing about CFTR modulators is that they're small molecules, and they can easily penetrate cells in vitro and in vivo, and they directly modify CFTR function. And what's cool as we get new and better modulators is we see that CFTR activity, as seen in human brachial epithelial cells in culture, so that's the x-axis, directly correlates to clinical outcomes, so change in FEV1 as seen in patients. So that makes it really easy for a modulator to develop a preclinical pipeline. We know what model to use, primary HBE cultures from multiple genotypes derived from patients. We know the assay to use. We're going to use electrophysiology and look for change in current. And we know the endpoint, right? % of wild type CFTR activity. And we know that if all those things line up, we're going to see a good benefit in the clinic, or we're pretty confident. Not 100%, but We've seen this time and time again with modulators, and this is why theratyping has been so successful. So what about with genetic therapies? Well, the problem here is that to date, we have had no durable clinical success from a genetic therapy for CF. So we don't know if there's a correlation between HBE ALI cultures treated with a genetic therapy and clinical outcomes. We just don't know. Will they predict clinical success? Maybe, but genetic therapies aren't the same as modulators. So there's a structure of ivacaftor versus a short string of RNA nucleotides. These aren't the same. Genetic therapies are big, they're bulky, they have to be delivered in a delivery vehicle, and they're diverse. They are not all the same. We have RNA-targeted approaches, antisense oligos, read-through inhibitors, mRNA replacement therapy, which we heard a lot about in plenary 1, and then DNA-focused approaches, gene replacement, and then finally, one day, editing and insertion. And each approach has really distinct strengths and barriers to success. You can come up if you want. You did such a good job. So I thought it might be helpful to think about what's happened with genetic therapies for other diseases. Examples of ASO exon skipping, Exondys 51 is an FDA-approved ASO for Duchenne muscular dystrophy. It's a weekly IV infusion. It results in a truncated but functional protein. And this is an example of a success. But DMD has a really long half-life, and it's expressed in non-dividing cells. This is distinct from CF. Examples of mRNA therapies, well, we've got COVID vaccines, like Becky Dara told us yesterday. We've all taken an mRNA therapy. 2 IM injections, boosters as indicated, and they target immune cell antigen recognition processing. But a functional protein, a functional transmembrane chloride channel is not required of an mRNA vaccine. So not quite the same as CF. And finally, gene replacement. Zolgensma is an FDA-approved therapy for spinal muscle atrophy. It's a single IV infusion of an AAV9, adeno-associated virus 9, containing the SMN1 gene. It targets non-dividing motor neurons, and SMA is a disease that is fatal by age 2. So the disease severity there increases the tolerance to risk for a genetic therapy. Again, differs from CF. In CF, we're trying to correct CFTR in tissues that divide and will require redosing. We need to produce a functional transmembrane chloride transport protein, which is not an easy challenge. And the risk-benefit ratio is really different. We have so many therapies that people with CF in general are doing pretty well. So the thought of a risky gene therapy, gene editing, is kind of difficult to swallow unless we are very confident that will be successful. And so we've taken all of these things into consideration as we've built a framework for a preclinical package development in CF. So what's the goal and the complexities of genetic therapies for CF? So the goal is to deliver CFTR, RNA, or DNA through the mucus barrier to the airway epithelium. And I specify that because all of the programs we're currently supporting through the CFF are targeting an inhaled therapy. We may one day see something that is systemic. Oops, I went. But for now, these are all inhaled therapies that will have to make their way through the mucus. And the goal is restoration of CFTR expression and function that will lead to rehydration of mucus and improvement in lung function. So what are the barriers? What are the complexities that we have to overcome? I'm showing mRNA therapy here as an example. Here, maybe I'll use the pointer. Oh yeah, okay. I'll show mRNA therapy here as an example, and we have barriers like cellular uptake through thick mucus. Once it gets into the cell, we have the barrier of endosomal escape, proper translation, protein folding and trafficking, and then chloride transport. But we also have, my animations don't work. Oh, there we go. We also have the complication of uptake by the wrong cells, movement of cilia that could move or trap genetic therapies, development of neutralizing antibodies, chronic infection that could impact the inflammatory and immune response. We have low pH that could affect stability and degradation of therapies, and cellular turnover. So I think it's apparent that there are a lot of barriers to overcome. And an ideal preclinical package would demonstrate that a genetic therapy can overcome all of these barriers. So if I put them all together, what we did is decide to group them. So there are barriers to delivery and distribution of the therapy, barriers to CFTR detection and gene expression. The fonts are super weird on this, I'm sorry. Barriers to detecting CFTR function, physiology, and then redosing and immunology. And so these are the major categories that we used to build our preclinical data framework. And then we asked for each of these areas, these buckets, what are the questions that we need to answer? So with regard to delivery and distribution, I think the number one question is, does the vehicle, we don't need to think about cargo, but does the vehicle get into the right cells? Does it get through the mucus? And how much gets into the correct cells and organs versus off targets? So you can test this in primary cells, wild type or CF, and also in vivo. And we can also look at stability of the vehicle in mucus. And there are many assays you can use that include reporter expression, IF, Western blot. And I think the endpoint we want to see is the percent of cells that are positive for expression of a reporter here. We don't even need CFTR. At this point, when we're just talking about delivery and distribution, a reporter is great. Then we get to CFTR detection. And we want to know how much CFTR is expressed compared to wild type. And I'm going to highlight the compared to wild type because there has to be a control. We want to know, does CFTR express in the right cells? We have a lot of new data that tells us CFTR is expressed in secretory cells, in ionocytes, and we know, or we think, that it matters what cell CFTR is expressed in. If it's in the wrong cells, it may not be functional. So we need to know, is it expressed in the right cells? And we also want to know how much CFTR is expressed in off-target cells and organs. We don't know that it'll be a problem, but we need to measure it and be aware. So here, again, we can use our primary wild-type HBEs. We can also use in vivo models. And we recognize that using CFTR null background can be helpful in detecting CFTR because we know that antibody staining and antibodies are challenging. And I think the endpoint that I want to emphasize here is the ratio of therapeutic CFTR, so the ratio of your rescued CFTR expression to wild type, as well as percent of key cells that are positive. So we want to know both. How many cells are positive and what is the level? With regard to function, we want to know, is CFTR dependent current restored? And I think this, for most of us, is the gold standard. We want you to do the electrophysiology. We also recognize that organoid swelling is a reasonable way to look at CFTR function. And this is a lot easier to do in cells and organoids than it is to do in vivo. But I would encourage you all, if you're developing genetic therapies, to think about doing this in ex vivo tissue that was treated with your genetic therapy. So again, we want to see a change in current, and we really want to see it in comparison to something, to wild type, to F508del treated with a modulator. If there's no control, if there's no comparison, it's not particularly helpful data. So the next bucket is physiology. Can therapeutic delivery improve physiological signs and symptoms of CF? And how does restoration compare to that of clinically validated modulators? And I think this space is a little bit more difficult, and I think Wanda and John are going to touch on this in their talks. This is one where we know that FDA does not require evidence. So if we ask for it, we're setting the bar pretty high. If we say to get into a clinical trial, we need preclinical data showing improvement of CFTR physiology, that's a high bar. But it's a high bar because we have so many drugs in our pipeline and we have so few patients. So we need to be really confident. So I'm not going to say that this is required, but we really, really encourage you to explore the models and tools we have and think about CFTR physiology in developing a preclinical package. And finally, redosing in immunology. So I pointed out that in cells that cycle, redosing of a genetic therapy is going to be required for a transient therapy, not possibly for editing. And so this is something that needs to be considered. And I'm showing this image here, and this is immune response to SARS-CoV-2, which is not a genetic therapy. But I'm showing it because I think our initial thought is, hey, neutralizing antibodies. We know about neutralizing antibodies. And my point here is that the immunological response to genetic material is a lot more than development of neutralizing antibodies. You have a lot of things going on that need to be detected, and we need to think as a group about how to analyze this in different in vivo models. So the questions we want to ask here are, does repeat delivery cause an inflammatory response? Are neutralizing antibodies detected against the vehicle and the cargo? And does cellular immunity contribute to loss of CFTR expression over time or expression of a transgene? And these assays, by definition, will have to be done in an animal with CF-like physiology. So what models and tools do we have? And this will just be a brief highlight because, again, Wanda and John are going to talk to this. But luckily, we have a lot. We have a whole zoo, and also we have these great HBE cultures in vivo. So I just highlight differences in their CF-like electrophysiology properties, because that was one of the points I highlighted. The diversity of genotypes available, the accumulation of CF-like mucus, immunology defects, hyperinflammation, survival requirements, availability, and cost. And we recognize that each of these animals really has its niche where it can be most helpful in developing your preclinical package. So what does our guidance look like today? With the Genetic Therapies Working Group at the CF Foundation, and in working with my co-chair, John Sheridan, who's in the audience here, we put out a very generalized guidance for sponsors developing genetic therapies. And we said, show us evidence that you can, the colors are all different. It's really throwing me off. At the bottom, show us evidence that your vehicle and reporter gene can get to the cells that you're targeting. In blue, show us evidence that the therapeutic CFTR message and protein are expressed. Show us evidence in green that chloride transport has been increased as a result of your therapy. Show us physiological improvement consistent with changes in CFTR function, and show us that this is safe and can be redosed. And combined, assays from those 5 buckets will give us confidence to push this product into clinical trials. And so we give this guidance to sponsors. We give it very early in CFF meetings. So this is when the foundation interacts with sponsors as they're building their development plan. We say, hey, these are the things to consider, because when you get to the clinic or when you want to get to the clinic, this is what the TDN is going to ask for. They get it again when they are referred to the TDN and they meet with the TDN, and the TDN says, hey, this is what we want to see before you go into clinical trials. And then when their protocol is ready, these genetic therapies will undergo what we call compound review. And that's a series of experts that will look at their preclinical data package and give a thumbs up or thumbs down. Is this ready for the clinic? And guess what the compound reviewers use? That same preclinical guidance. So my advice is listen to the guidance. And I'll say that at the beginning, we were not very prescriptive. When we put this out, we were very general. And we said, hey, there's lots of assays, there's lots of ways you can choose to do this. But as we see companies moving forward and developing preclinical packages, we know we have to be a a little more specific. So looking to the future, we're revising our guidance. We're revising it already. So at the top, with regard to delivery and distribution, we initially said, give us evidence that your thing gets to what you're targeting. And we saw people detecting markers on their vehicle. Well, sometimes markers fall off. Labels, fluorescent labels, they fall off. We don't want you to label the vehicle. We want you to deliver something that expresses a protein. We want you to use modulator unresponsive genotypes if you're going to do this, because those are the first people that are going to get your product. We want you to quantify delivery and distribution. We don't want to see just one really pretty microscope image. Those are great. I love microscopy. But one picture does not convince me that your vehicle got into all the cells it needs to get into. So quantification is really important. And controls. Controls are really important. Empty vehicle, super important. So anyway, I'm not going to go through all of this, but the point, and I'm happy to share it with whoever wants, the point is we are standardizing the way preclinical packages are built so that by the time these groups get to the TDN, by the time they are ready to take this into the clinic, we have a standardized set of assays that we can compare. And we can say, A looks better than C, but C looks better than G, right? We can put the best packages forward for that small group of patients who are available. And so with that, I will wrap up. And Wanda will come to the stage and talk about small animal models that can be used in developing genetic therapies.
Hello, thank you very much for the invite to talk. So I was asked to talk about small animal models in the context of this environment and where I'm at. Small animal includes mice, rats, and rabbits. And John will talk about ferrets and pigs and maybe sheep. So the question is, how do you heard Kara say that we need some functional data, we need some evidence that your products are working in the clinic. How can small animal models fit into that research plan? Oh, goodness. Okay, so here's my disclosure. I do get a little royalty when the beta ENaC mouse is used, so anything I say about that, take with a grain of salt. So small animal models will be used in the pipeline for any drug that goes into people, right? So even if it's later on in the development of the drug, eventually you'll have to put your drug through a toxicology screen in models. Rat, rabbit, non-human primates, dogs, mice are commonly used for biodistribution and toxicology studies. But in CF in particular, we have this really cool option to use a multiple of models to actually study function. And and so the cystic fibrosis models that you have access to as researchers working to develop a drug are the results of a huge amount of effort. And so how did we get to this point? So CFTR orthologs are expressed in vertebrates from cartilaginous fish onward. And because of all the technology to make mutations in these different species, this has allowed the development of multiple CF models with the numbers growing. And when I did a little pub search, typing in cystic fibrosis in animal models, you can see that, after the CF gene was cloned, there's been like an exponential increase in at least these two terms being used together. And so animal models are having a big impact in the way we think about CF. And that, that's just because, since 1992, we have developed all of these models. And these are all described in the literature. There's a lot of nice reviews. Here's a couple of the more recent ones that give a lot of the references if you need to reference these animal models and their characteristics. These papers have great references. And so there's just this huge opportunity to use these models to support your work. Now, notably, none of the currently available modulator therapies directly stem from model research. So as Kara was saying, it's like sort of you have this balance now of trying to decide when do you really need to use a model. And what she's suggesting is that the guidelines are becoming very specific and that there will be a requirement for some in vivo studies in order to efficiently move your product forward, at least, working with the CF Foundation to do so. So the animal models available, as Kara already suggested, have different advantages and disadvantages. So here is the homology of the amino acid sequence to human. And here are some of the disease features of the human model, our best model, of course. And as you can see, as you sort of move away to these small animal models, you lose some of the important features. And so as you're developing your model, you have to decide, is the loss of these features going to matter to my program? But it's also important to notice that all of these animals have functional CFTR loss. So if you're looking for correction of CFTR function, you can measure that in all of these models. And so then the main consideration, wow, something exciting is happening over there.
Wow.
They're clapping for me, yes. So the main consideration is sort of this balance then between cost and utility. Of course, the mice are going to be the cheapest. They're going to be able to use those in the largest number. And then the rat, then the rabbit, and these other models are going to be really quite expensive to use because of their long lifespan, et cetera. But they will have an increased human-like CF physiology. And so the balance between that is important, and it will have to be figured out in each developmental program. And so it's like, well, maybe for one part of your program you'll be able to use the mouse, but another part of your program you'll have to move to the pig, right? It depends on the question that needs to be asked to address your problem. So Kara talked about this pipeline, and I'm just showing it in a little bit different way, from delivery to distribution. There's many considerations. And luckily for us in the CF community, there's assays in these animal models that can address all of these issues. We can look at delivery, we can look at detection with these assays, we can look at CFTR function. In certain ways, we can establish physiological correction. And we certainly need animal models to establish whether we can redose, because we need an active immune response in order to understand those very complex processes. So animal models can be used effectively for all of these things that Kara talked about that will be needed to support therapeutic development. So you have all these questions. There's lots of questions. There's lots of models. Are you using the right model? This little mouse would say, me, me, me. So it's difficult. It's a difficult decision. And again, it's a balance between cost and what you need and your expertise and your experts. And, trying to think about what mice and rats and rabbits might be good for versus what other models might be good for. I think they're very good for proof of concept. They're definitely good if you feel like you need a large statistically powered study. You would not be able to do that in large animal models. They're going to be really good for screening non-viral vectors. They're not going to be so good for screening viral vectors if you need host tropism to be the same. So you can't expect an AAV vector that's specific for mouse airways to work in a human, right? It has to bind the receptor in human, not the mouse one. And so that has to be considered. They're very good for biodistribution studies or reporter screens. They're also good for intestinal biology. All of the models, mice, rat, and rabbit, I'll show you a little bit more of that later, develop intestinal features. And although Kara says that all of the drugs coming through the pipeline right now are aerosol delivery to the lung, intestinal problems are important for CF patients, as are pancreatic problems. And so it's not something that we as a field should ignore. And some of these features of these models will work for those phenotypes. They're also they're going to be useful for you for functional correction and electrophysiology, and of course toxicity and immune responses. So I want to just spend a minute to talk to you about functional correction, because I think this is where the models really are useful in the mice and rat and rabbit. And of course, one of the ways that people look at this is nasal potential difference. This is just a picture from this paper that utilized this assay, which was first developed by Mike Knowles and his colleagues in the human. And you can use this assay to detect differences in a living animal between CF mice, which don't respond to low chloride, and wild-type mice, which do. And this is a sensitive assay. It's got a good dynamic range. And there's many, many examples in the literature of where this assay is being used successfully. And I think that if you were able to convince the CF Foundation that you had a good response in nasal potential difference, it would go a long way toward convincing them that your agent might be ready for human. Just as an example, just from this conference, Alexandra McCarran from Australia shared this with me. It's on her poster number 629, where she used a lentivirus gene transfer vector in neonatal CF rats. You can see the expression of a beta-gal vector, and then with a CFTR vector, you can see a change in nasal potential difference toward the wild-type level after the vector treatment. So this is an example of functional correction. Now, the small animal models can also be used to study other robust non-respiratory phenotypes. The intestine is particularly good at this. It has a very robust electrophysiological profile that distinguishes between wild-type and CFTR. It has a robust histological profile. The mucus plugging is pretty obvious in the CF intestine, not so much in wild-type. Here's a rabbit appendix, and this was one of the very most robust phenotypes we saw in the rabbit. They have a huge problem in their appendix. And so you could imagine that a systemic therapy designed to treat, am I doing with time? This would be a very sensitive and quantitative way to measure whether or not you're making a difference, is to look at the rabbit appendix. Survival is also a phenotype that is robust in these animal models. Without treatment like cholite to keep the intestinal obstruction at bay, there is very low survival. This is an example from rats. If you give the PEG, polyethylene glycol, survival is increased. Well, if you saw this kind of increase with a treatment, you would be very excited. And interestingly enough, there's very simple CFTR phenotypes that can be measured, including the development of this enamel defect that you could theoretically monitor. Okay, and then when asking is CFTR being delivered to the correct cells, what models? So the important thing to take away from this slide is that new molecular characterization is starting to add knowledge. So single-cell sequencing, this is just one example of single-cell sequencing from multiple species. And we're really beginning to understand truly the differences in cell types across species. And what we know for sure is that no small animal model fully recapitulates cell types in the human lung. So that is an important consideration when you're trying to figure out whether or not you're targeting to the correct cells. For example, basal cells are not present in the mouse small airway. Mice and rabbits lack submucosal glands. So if you're trying to target submucosal glands, mice and rabbits are not a good model. Mice, rats, and rabbits lack respiratory bronchioles. Respiratory bronchioles are maybe thought to be a very important target for human CF. And in fact, mice lack bronchial circulation. So if you think your vector needs to be delivered to the airway via bronchial circulation, you might have some troubles. So, and then finally, what about the gene correction strategies? So there are many, many different ways you can think about gene correction. We've heard them all. There are some that are very specific to mutations. So the nonsense mutations, the splice site mutations, the premature stop mutations, what kind of correction are you aiming for? And for this work, there are many, many models potentially useful for these purposes that are now available that should be used for this purpose to support your program, especially in the mouse. And I just highlight the Case Western Reserve University CF Mouse Models Core. They have a poster number 640 in collaboration with their transgenic and targeting facility. They have a wide number of alleles available from the mouse CFTR, and importantly, now also a wide number of humanized variants. I don't have time to go through all of these, but if you need a specific correction strategy targeted to a specific sequence, there are actually models in mouse available for you to study that. How's my time? Okay, I have time. Okay, and then so what about the rabbit? So the rabbit is a little bigger. Some might not consider it a small animal model, so it has some unique features because of its size. And one of the things that our group has been doing at UNC is to take advantage of the size to develop this rabbit tracheal explant model. So in this model, the trachea is dissected and opened up and placed on a gel foam matrix and grown in culture, and it can stay alive for several weeks. And so this is an example of what the explant culture looks like after 7 days versus the in vivo trachea, and you can see that on this gel foam matrix, the histology and the cell types and the morphology is very, very similar. This is what it looks like when it's growing on this foam. And with RNA in situ hybridization studies, we've been able to demonstrate that this tracheal explant from the rabbit trachea expresses high level of CFTR activity. This is a, I'm going to try to get this to work. Oh, darn, it's not going to work. This would show cilia beating. Oh, it is showing it. Cilia beating and beads moving, which tells us that this explant is functioning normally. The mucociliary clearance in these explant cultures is maintained up to 21 days. And so we are using this as a complement to human culture studies. So in these explant cultures, we can demonstrate that from the CF rabbit, the expected loss of CFTR-mediated current compared to wild type is seen, and that we can utilize this explant culture to try to now start understand when we start to deliver things to the surface of these explants to help us predict what might happen in the actual model. This is an example of a Sendai GFP expression in the explant model, an example of electroporation of GFP into the explant model as well. So the rabbit with its little bit bigger size provides this opportunity to develop this explant model and to use it effectively. And finally, for the mouse, another plug for mice, is that there are non-CFTR related models that are useful for this kind of program. And in particular, the beta-ENaC model for mucus penetration studies. So we've all been hearing that if you're going to aerosolize a drug, it has to get through the CF mucus and it has to get to the airway surface, it has to get to the cells that you're targeting. The beta-ENaC model produces a mucus plug, viscous, sticky mucus, because it hyperabsorbs water because of overexpression of the sodium channel. And this is described in some of these papers. And as an example of how drug companies and others are using this model, I present this work that was presented in Thorax very recently, where the company utilized two different kinds of particles to deliver their cargo. One of them was large and positively charged, which you can appreciate did not make it to the airway surface, versus a mucus penetrating particle, which was less than 50 nanomolars and negatively charged. And that particular formulation made it onto the airway surface of the beta-ENaC mice, and suggesting that it makes it through the mucus. So here's the mucus penetrating particle making it to the airway surface versus the non-mucus penetrating particle. So if you want to demonstrate that your drug or your formulation can make its way through mucus, the beta-ENaC model, which is a small animal model in mice, can be utilized effectively, and it has been utilized in this way. So mice and rats and rabbits have lots of good things going for them. Functional assays are available for all of them. For mice especially, there's multiple CFTR variants, especially those that can be used for these very specific mutations that you're trying to target with these prime editors and these base editors. Those mice are available, and they should be utilized to demonstrate functional studies. Of course, the lung disease is minimal. In these models, they have problems, and maybe they don't have submucosal glands or they don't have the right cells. They all develop gut pathology, so this can be a good thing if you're trying to understand systemic delivery. It can be a bad thing if you're trying to keep them alive. So that's why it's in yellow here. It's sort of a caution. And of course, none of these models are human. And so they'll always have to be interpreted in the context of they are a model and not representing the human situation. So here's my acknowledgments. Thanks to Kara and Deepika. Thanks for all these folks who shared their data and slides. And I'll take questions during the panel. And I welcome up John. Thank you.
Oh, it is? Okay, perfect, perfect. All right, thank you, Kara and Deepika, for inviting me to speak. I do have some disclosures. Some of these are relevant, what I'm going to speak about. Let me get my pointer here. I'll be talking about some research with both Spirovant and Arcturus that's sponsored. So like Wanda pointed out, we're extremely lucky as a field to have multiple animal models. The phenotypes within these models are quite diverse. Currently, the ferret, pig, and sheep are ones that reproduce these organ pathologies, with the exception that the sheep has yet to be reared to adulthood to study lung disease. I think it's important to point out when thinking about the lung as a target is that CF is a multi-organ disease, and disease in the other organs can impact disease progression in the lung. Now, ferrets and pigs develop pancreatic disease as well as lung disease here at adulthood as compared to what Wanda had already presented in mice. Sheep develop pancreatic disease, CF sheep. At birth, their lungs are normal. That's very similar to ferrets and pigs at birth. But there are hints that the CF sheep would be a good model for lung disease. You can see this gland plugging here, which is also seen in a more exaggerated fashion in older CF pigs and ferrets. Now, I think it's impossible to talk about lung as a target for gene therapy without thinking about the differences in cell biology and anatomy between the various models. Pigs, ferret, sheep, and human have submucosal glands throughout their cartilaginous airways. These are associated with ionocytes that we've heard a lot about at this meeting, and a secretory cell called goblet cell, which is slightly different than secretory cells that exist in the bronchioles, Clara cells for humans and ferrets and pigs. Now, mice have glands in the proximal trachea. It's a viable model to study cell biology, but it's not viable to study conducting airways disease in CF. They also have a small number of ionocytes, but their primary secretory cell type throughout the airway is the club cell. Now, the other difference that Wanda touched on is distal airway biology that's different between the smaller species compared to the species listed here. So the bronchioles, like I mentioned, primarily composed of club and ciliated cells. There are more basal cells than you find in the smaller species. But I think it's important to point out that the respiratory bronchioles, which are becoming of great interest from a stem cell biology standpoint, from some leaders in the field, have club cells that are interlaced with alveolar cells, almost in a fenestrated fashion like you see right here. This region expresses significant amount of CFTR. And I'll point out that all of these cells express CFTR. And as mentioned at some of the other meetings, we know nothing about what alveolar type 2 cells are doing, and cumulatively they express most CFTR in the entire lung when summed up. So pigs, ferrets, or sheep, mouse to pigs, what are the differences? I'll just sum it up here. Pigs, ferrets, and sheep are the closest to human. There are some differences in developmental biology of the airways, but are largely a better representation. And CFTR is expressed in each of these regions, as I mentioned. Now, one of the biggest hurdles is cost. Here I've normalized this to a mouse at 1x, and you can see cost increases with size, as one might expect. And this is a challenge to study design and some things that I'll talk about. So submucosal glands may be an important component of modeling CF airways disease, and really they're only present in the larger animal models. And rodent interpulmonary airways really model the distal airways in humans, and both in size as well as in cell type compared to the larger species. So what are the limitations of the larger animal models? Well, the frequency of meconium ileus is one. With the smaller animal models, they at least are rearable prior to weaning. With larger animal models, meconium ileus truly models the CF defect. There are some ways to get around this. There are gut-corrected pigs, CFTR modulator responsive pigs, ferrets, and sheep. But the cost, which is significant, scales with the size of the animal. Us at Iowa, we're working on developing conditional knockout, whole body knockout ferrets that are in the pipeline. Another limitation is study power. You know, often companies will come to us to collaborate with the mindset of a mouse or rat study, and that's just not feasible in the larger animal models. You know, most of the studies that I prefer are an N of 1 crossover study where you're comparing the responsiveness to a CFTR modulator in response to some therapy and doing baseline studies. And there's smaller number of animals. This often will impede gender, you know, having equal genders in your studies because there are some things you just can't control when the animals are born. So CF ferrets we've developed CF ferrets. I'm going to focus for the remainder of my talk talking about what's possible in ferrets. The first model we developed was CFTR knockout. We've since developed a ivacaftor VX770 responsive CF ferret, also develops lung disease, and if maintained on the modulator, is free of most disease. It's not completely protective, but very close to full protection with a clean knockout at the CFTR locus. These models develop bacterial infection when removed from modulators within the first 2 to 4 months of life. And the most value from this model, at least in my opinion, is not only an easier way to rear and bulk up on larger numbers to do the studies, but it's also to study the disease onset as well as the reversal of disease processes that we're trying to treat by gene therapy. There was talk yesterday about Alice and Lance, and that talked about Pseudomonas infection in these models. We didn't realize that they would become infected with Pseudomonas until we had this very old animal through COVID time that had managed to develop Pseudomonas, mucoid Pseudomonas with a very similar mutation to humans. And since then, we're learning how to try to speed up this process. So I think about gene therapy, 2 major problems in gene therapy that relate to CFTR function or stem cells and gene editing. From a CFTR function standpoint, what cells must be corrected to complement the disease phenotype, and what cells have the capacity to complement the disease phenotype, even though they may not normally express CFTR. One of the things that I'll be talking about are approaches where we're trying to tackle this question genetically in ferret models to give a pure answer to the question. And then second, if you're using a stem cell gene editing approach, what stem cell compartments can differentiate into CFTR expressing cellular compartments? And there are likely going to be many because there are at least 4 independent trophic units in the lung that have independent stem cell compartments that will need to be targeted. I'll touch on a few of these aspects. So we've developed over the past 20 years technologies to be able to genetically engineer ferrets using a number of approaches that Cas9 injection into zygotes, either by homologous recombination or non-homologous end joining to create types of ferret models that can be applied in the below blue highlights. And I'll touch on some of these. The basis of really being able to ask sophisticated questions like have been addressed in Mitch Drum's group with mice and is conditional genetics. This, for those of you that don't know, there are a number of recombinases that can target sites, very long base pair recognition sites in the genome, and if fused to an estrogen receptor, can limit the presence of that recombinase within the nucleus. You give a prodrug, in this case tamoxifen, mobilize it to the nucleus, and this can excise or flip sequences within the genome. So this is the basis of conditional genetics and allowing to control, either activate expression of CFTR in any cell that you want or inactivate it in a temporal fashion. So one of the long-term goals of my group is to be able to ask what cells are required to have CFTR expression and what cells can complement CFTR expression. And one of the approaches, for example, in this wild-type airway is to delete CFTR cells specifically within cells that express endogenous CFTR, or to reactivate CFTR expression within those cells. And likely this may have to be done in combination because it's not as simple as any one cell type being responsible for the pathology in CF. I'm going to just quickly touch on a component here where we've tackled this to address ionocyte function that I spoke on yesterday. So we've created 3 different genetic models. The middle one here is one that expresses the recombinase specifically in ionocytes under the direction of a gene that's only expressed in ionocytes. A Cre reporter that switches from tomato to EGFP when this is deleted by Cre, and then conditional CFTR knockout ferrets. And so we can lineage trace ionocytes if we induce double transgenic animals with tamoxifen, and you see them here in green. And if we put this genetic conditional knockout background on the same animal. You can delete CFTR only in those cells and ask functional consequences of that deletion. So if you're interested, just search ionocyte on the web and you'll find all these talks, and hopefully they're all online because many of them were yesterday. So another application of these genetic ferret models are to study stem cell biology. Here's an example where we're studying an organ very close and dear to my heart, submucosal glands, where we have a Cree driver that's labeling a stem cell compartment, which incidentally we learned about in mice, but we can't study CF airways disease with that. And then are studying in the context of CF in ferrets. So here we've lineage traced myoepithelial cells around these gland tubules and smooth muscle cells. And here's an example asking the question, is this a compartment that would be suitable to target by gene editing? So we injured the airway of animals that were induced with polydocanol and harvest them later. And then you can see here a picture of a gland duct leading out to the surface in two different planes, these EGFP positive cells emerging on the surface. And basically this is just a phenotyping of cells that are in both the glands as well as on the surface, demonstrating that we can target the myoepithelial cells are precursors for basal cells on the surface, secretory cells, as well as ciliated cells. So these glandular myoepithelial cells may be a viable gene editing target for the correction of CFTR, both in the glands as well as in the surface airway epithelium. The last example, or another example that I like to give, is utilizing the transgenic reporter ferrets to index the efficiency of gene transfer in a digital way that's very robust. So we're interested in using lipid nanoparticles for gene editing in ferret models. And we teamed up with Arcturus, who was interested in applying a CFTR mRNA therapy. Some of the first studies we did were to test Cree mRNA within this lipid nanoparticle within the Cree reporter ferret that turns red to green. And so I want to just sort of outline what the pipeline is at the resource center that manages the ferrets and access to the ferrets for evaluating gene therapy vector and that experience with Arcturus. So the first question I always ask, is there anything that's species specific? I mean, it's a lipid nanoparticle that's got some ligand on it that has the potential to be different from the organ than the species that it was developed in. It may not work in ferret, so you've got to test that first. Using something robust like a Cree reporter. Here's the surface of an air-liquid interface culture that was transfected apically with lunar Cree. You can see about 30–40% of cells were transfected and converted to the GFP reporter. The next is functional, like Kara pointed out. Compare this to some metric where you have an index of efficiency of CFTR correction, electrophysiology. And here's an example with increases in concentrations of CFTR lunar against VX770 and G551D ferret air-liquid interface cultures. Then you're ready to go in vivo. And so this is an example of using the lunar Cree in vivo in a Rosa TG ferret, a fairly efficient gene transfer. You can see the green here in the surface airway, in the interlobar airways, as well as in the trachea. You can also do this with another reporter. Here is tomato mRNA, where you can see fairly efficient interlobar transfection and expression of the tomato. Then lastly, it gets into the more complicated and difficult experiments, and we're not through all of these yet, but I'm going to give some experience about this. Questions like, can the vector that you're using get through the CF mucus? And ultimately, can it reverse an endpoint? So here's an example. I just showed you some wild-type animals on the previous slide. So here's an example of a mild CF disease ferret about 5 months old off VX770. You can see mucus accumulation here in the airway and fairly efficient transfection to the trachea, maybe a little bit reduced in the intrapulmonary airways. But here's an example of a ferret off VX770 for 9 months, completely occluded cast here with maybe a lumen that's allowing access to the transfection material out into the alveolar regions of the lung, and likely significant cell death in the mucus as evident by the green accumulation there. So this is the major problem, and this is a problem for any vector. airway plugging and mucus casts will be impenetrable to any vector system. The solutions are mucolytics. We've been working on that, but I won't have the time to show you that because I want to present a couple of other gene therapy approaches. Other approaches to gene therapy include RNA viruses, lentiviruses, retroviruses. Here's a case where Paul McRae and the CF pig used an FIV, feline immunodeficiency virus, transfecting CFTR, fairly good correction, pulling out airways. Here's an example in a ferret using the same FIV vector pseudotype with GP64. It's highly effective in interlobar airways. We've also been evaluating vectors for transducing the pancreas. Here's an example using an EGFP vector stained for insulin after IV injection in a week-old ferret. Lastly, I do want to touch on some studies that we're doing and were touched on by Kara about immunology and the importance of beginning to understand that when testing viral vectors in vivo. This is a study that was done with Spirovant, where we evaluated repeat dosing of AAV to the ferret lung. The first dose was all self except for the capsid, ferret CFTR with the deleted R domain CFTR. The second dose, 4 weeks later, was with Gaussian luciferase, which is a very robust endpoint for gene expression, and then harvested at a relatively short time point of 2 weeks after that. These are results of expression of the entire lung, pulverized lung, so cumulative expression, avoiding any distribution problems in delivery of the vector. And what you can see here is that single dose, the repeat dosing is about 10-fold lower as far as gene expression, and the repeat dosing is about 5-fold higher in neutralizing antibodies, which correlates with reduced efficiency. So repeat administration of this vector, which is AAV2.5T, within a 1-month window is limited by neutralizing antibodies in the lung. And there are several questions that this raises. One, how long will those neutralizing antibodies persist? And are you going to have to repeat at a frequency that will be inhibited by that based on the durability of transgene expression? And the second is, can you use approaches to limit this neutralizing antibody response? And so I'm going to go through this quickly because I'm running up at the end of my time, but this is an experiment where we compared 2 different immunosuppressive strategies. This tri-immunosuppression is what lung transplant surgeons use in CF patients that receive a new lung. So this is like the hammer, and then we tried methylprednisolone alone. The animals were treated 2 days before they received the first dose, which was ferret CFTR, and then 4 weeks later received the Gaussian luciferase, and then sacrificed again 2 weeks after that. So what did we see? Well, neutralizing antibodies, surprisingly, between the tri-immunosuppression or methylprednisolone were no different, but they were lower than repeat dosing in the absence of treatment. And the transgene expression was higher with methylprednisolone, and with the tri-immunosuppression was about another 10-fold higher than that. What was interesting, if you compared to a single dose of virus on the previous slide, that's right about here. So there are some unknowns that we don't realize yet, that this triple immunosuppression had inhibited some aspect that was limiting gene transfer, but it wasn't neutralizing antibodies. I'm just quickly going to go through efficacy endpoints are really important. We've hammered really hard using pulmonary function tests, and I'll just sum this up by saying ferrets, you don't have a predictive FEV. There's a very large sexual dimorphism in size, so you need large numbers to get that sort of predicted FEV. And just summarizing pulmonary function tests as a whole are okay for detecting very chronic changes, mostly with inspiratory capacity and forced vital capacity. The most robust endpoints that we've had have been with mucociliary clearance, and Kara showed this. I'll just quickly go through it. We're just comparing CF animals that are on VX-770 and off here to a wild-type animal. The most informative data is here on the left, where you're comparing a scan of clearance in the trachea. Then the animals are taken off, the X770 progressively goes down and put back on for 2 weeks, and then it pops back up to normal. So, I mean, this is the type of pattern that I think we can have a robust endpoint as an index for gene therapy, knowing that this clearance is CFTR dependent. So to summarize, I think genetic ferret models can aid in the dissection of CFTR function at the cellular level, either through conditional activation or knockout. And genetic ferret models capable of lineage tracing, I think, could be valuable in both identifying the stem cell compartments, but also evolving viral vectors using Cree conditional genetics to barcode infrequent capsids that are able to transduce that compartment. And then the ROSA TG model I showed you is fairly robust. I think Mitch is working on a number of approaches to index gene editing, and I'm anxious to see how those work and maybe transition those as appropriate into a model like the ferret. And mucociliary clearance is the most robust endpoint we have now in vivo following gene transfer, as the pulmonary function is mostly detecting very chronic disease states. Thank you.
Thank you, John.
Hi, I'm Deepika Polaneni. I'm an associate professor at Washington University in St. Louis. I am the other co-chair with Chara. Thank you for covering, Kara. And I'll be speaking today on key biomarkers for proof of concept human studies. I think it's funny that I'm the only person not talking about animal models because my heart is still going like a hummingbird after being in front of thousands of you. So please pardon me if I'm a little out of sorts. These are my disclosures. So I would never ask anybody to read all the text on this slide. It was intentionally overwhelming because for me, as I started my junior faculty portion of my career, this is what it felt a little bit like the onslaught of text was, as people were really talking about discovery of biomarkers. And so I think it's apt that the US FDA and NIH put together the Biomarkers, Endpoints, and Other Tools, or BEST consortium, to help define and standardize these terms so that we can be more organized and moving forward on the same page. And so, according to the BEST definition, a biomarker is a characteristic that is measured as an indicator of normal or biological processes or responses to an exposure or intervention, including therapeutic intervention. And these are really categorized into molecular, histologic, radiographic, and physiologic markers. And actually, those are the exact categories that I'll be going through today. These are important for us to define because, as we just talked about a little while ago, the number of genetic therapies that are coming into the pipeline for trials for proof of concept and early phase studies is quite remarkable. That was covered by Dr. Steve Rowe in Plenary 1, and this is representative of just some of those that are in preclinical development, with one already enrolling underway. A key consideration before we move forward in discussing biomarkers is also the anatomic delivery and sampling of the same. So on the left, this is just a picture of some of the procedures or processes of determining nasal potential difference, which was really pioneered, as Wanda said, by Mike Knowles. I see Larry Johnson in the room, who also worked on this in early gene therapy trials. And on the right, standard bronchoscopy. If anyone is not familiar, this is a frequently performed clinical procedure in pulmonary medicine, less so in adults with cystic fibrosis, where we expect that these therapies might be tested, but still also in the pediatric population. And so when we consider looking at biomarkers, of course, we need to consider two factors. Where are we thinking about the dose delivery, and where do we expect to see the testing to be performed then? So the first of these areas that I'll cover is probably the most obvious: direct measures of CFTR and CFTR function. And so the first that we would really discuss is the messenger RNA PCR as a marker of CFTR. So this would certainly be more useful for DNA therapies than mRNA replacement therapies because intracellular mRNA must be assured, of course, to be clear that this is actually correct uptake and that you're seeing an effect of CFTR. This is advantageous in the sense that this can be batched and centralized, But the primer specificity may be, and that the primer specificity may be used to discriminate native from non-native CFTR if it's introduced into the sequence, so we can actually get specific into looking at the product. And this would likely be coming from endobronchial brushes on that bronchoscopy procedure that I just showed, although nasal sampling is also possible and well established from measures such as nasal curettage biopsies or perhaps brushes as well. However, one of the caveats is that mRNA expression certainly does not reflect protein expression, and that the reproducibility within a subject, I can attest from nasal curettage studies, can be quite variable, and can be also based in regional variability of sampling. So John just showed some beautiful immunofluorescence staining, and so of course we consider immunohistochemistry, immunofluorescence, and in situ hybridization. These would again be resulting likely from endobronchial biopsies or cryobiopsies, possibly brushes. And this would in fact be more apt to show us what types of cells we're actually getting signal from. Again, this would be advantageous in the sense that this can be batched and centralized, and cryobiopsy might in fact improve the yields, particularly of the surface epithelia. In situ hybridization, of course, can also then reveal to us cell types with increased sensitivity, but the lower end of sensitivity for these is unknown and likely inferior to PCR, and the antibody selection is certainly crucial and evolving. And then there's always the elusive Western blot in cystic fibrosis. So this is the in vitro gold standard, of course, for a processed protein, and it likely requires endobronchial biopsies or cryobiopsies again, not only brushings, however, because the yield would likely be limited in terms of what this could actually deliver for testing. This is somewhat advantageous because it could incorporate an epithelial cell marker to control for the biopsy death, and we've seen this. Also an example with rectal organoid and sampling for that. And again, finally, this can be batched and centralized. But there are several caveats to be taken into consideration, including poor low-end sensitivities, challenging backgrounds depending on the mutation, the fact that this is semi-quantitative and may not be distinguishing the surface localized CFTR from subsurface. And again, the antibody selection is crucial, and this is actually quite technically challenging for many labs. So nasal potential difference, this is moving out of CFTR detection to moving into functional detection of CFTR, was certainly established as the gold standard for measuring, or for direct measures of sodium and chloride channel function. And Wanda did show some very nice tracings of those potential differences in animal models. This is just one that's representative from a normal and CF subject from a paper from Dr. Rowe and colleagues. But this is actually something that I think that we are more limited now in being able to consider than we were in past eras of looking at gene delivery, as we're looking more at aerosolized delivery into the lungs. So that brings me really over to talking about lower airway potential difference. And this slide is courtesy of Marty Solomon, who really covered this subject more in depth on behalf of his lab and his participation in the National Resource Center and work with Steve Rowe and many other colleagues. So this is just a photo here of the apparatus that's used for their lower airway potential difference testing. This is actually the direct introduction of the catheter into which can make its way down to the second and third generation bronchi for direct visualization and installation of these measures to be able to look at changes in potential difference. And here on the bottom right, you can see some of those representations and what you might consider to be varying degrees of CFTR dysfunction that exist in a different lung disease and varying scale. So some of the challenges that we need to consider when we're thinking about potential difference measures both in the nose and in the lung is that these are subject to accuracy issues, both also for temporal variation and regional variation. There are certainly risks that are associated with bronchoscopy and sedation that must be considered for lower airway potential difference measures. Certainly, if you have small airways disease and cystic fibrosis and an instrument like this is introduced into those lower bronchi, it is going to elicit cough, which can make the measurements very challenging. And so the goal really is to then further establish normal NCF data for lower airway potential difference correlations to nasal potential difference values so that we can get a better reference understanding. And there's definitely a need to standardize and integrate these equipments so that these are capable of image-guided measures in both the nose and the lower airways. So that really moves us more out of the molecular and some of the physiologic, but we're going further into preclinical physiologic and radiographic measures. And I just want to say that sometimes you think you're standing on the shoulders of giants, other times you realize you actually are. Several of these slides are videos that were given by investigators who are experts in these areas, and I thank them for their contributions. So this slide is courtesy of Scott Donaldson at UNC, and this is a fantastic representation of what happens with mucociliary clearance imaging, which is actually just the clearance of radionucleotide-labeled particles that have been inhaled. And so this is a subject who has a G551D mutation that is being treated with ivacaftor. On the right, you can see that this is pre-drug, and on the left, post-drug. So just to get this out of the way first, the portion that you see post-drug that's going down into the right lower corner of that screen is actually a proportion that is coming up and being swallowed. And this is inert, so this is quite safe. But overall, what you see here is very good news. You are seeing the retention of that in the lungs because it's not being efficiently exhaled or coughed during the cough clearance portion, sometimes of these studies, and just retained in the lung, whereas in the left, you can clearly see this moving from the lung up the large airways and out. So this is on ivacaftor, which is a highly effective CFTR modulator. However, we know that by looking at graphs like these in several subjects who have been exposed to either elexacaftor, tezacaftor, ivacaftor, or trikafta versus ivacaftor alone, or the two-drug combination of lumacaftor, ivacaftor, which did have a lower clinical effect, we can see what we might consider to be correlative changes in mucociliary clearance. So you can see a higher level of effect with trikafta or ivacaftor, for example, and there are significant differences between these and patients who were taking lumacaftor, ivacaftor, again, where we did not see as much of a clinical effect. So you might think that this was maybe bad news to not see this change, but in fact, in the grand scheme of things, this is actually quite advantageous because as we move forward looking at the potential efficacy of gene therapy trials, or genetic therapy trials, I should say, this actually has helped us to understand that we can develop or establish lower limits of sensitivity and detection by experiences such as these with LUM-IVA. So this is just a general schematic to finish driving home the ideas of these mucociliary clearance studies. Certainly we can have hydrators that improve the airway surface liquid and mucus hydration, potentially also mucolytics that can be affecting the mucus rheology, and whether that's alone or taken together, these really do change the mucociliary clearance overall and or cough clearance, and we have seen these translate to clinical benefits with improved lung function and decreased exacerbation in the clinic. So the next tool that I'll talk about in the radiographic portfolio is high-resolution computed tomography. This is also something that we frequently see in the clinic in terms of the diagnosis of bronchiectasis, and so this is actually a cross-section of a mild bronchiectatic changes that you can see out here in the peripheral airways, also with mucus plugging in a patient with cystic fibrosis. So high-resolution computed tomography has been very well established, but we do need to standardize protocols and uniform scoring systems to be applied for these types of studies of early detection and biomarkers. There are low-dose high-res CT protocols that are available, and these have been used widely in lung cancer screening now, so we know how we can get these out in safer ways to patients for frequent monitoring. And we actually have evidence of high-res CT also detecting highly effective modulator therapy effects. The sensitivity to small effects or in shorter time frames, however, is uncertain. And the utility of this for short-term changes, especially for proof of concept studies, is certainly less well established. But we do have emerging scoring systems that may be more informative, that are more quantitative, such as the CF Pragma score. And that moves me from stagnant images, such as CT scans, over to more dynamic imaging. So these next slides are courtesy of Dr. Jennifer Goralski, who is really an imaging specialist now in MRI. And this is showing the dynamic breath-by-breath analysis of ventilation kinetics with a commercially available inhalation contrast agent. So on the left, what you can see is the 19F voxel signal over time. And these are actually with 5 breath holds. So this is actually quite different from when we do a single breath hold, which is just giving you a static representation of the activity in the lung. With 5 breath holds of this gas, you can see that you get changes that would be more reflective of what the dynamic stretch and ventilation is like that's occurring during normal breathing. So you can see this peak, and then there's a pretty quick expo. And on the right, this is a healthy lung, but on the left, you can see that with moderate CF, that is certainly more vibrant and delayed. One of the important factors for these types of imaging studies is being able to have agreement on the scoring and the standardization of the same, and the ability to look at these across multiple centers. So this is a nice study that's summarized here across 2 sites in Toronto and Cincinnati. This was a study of CF in healthy adolescents, and it's the first to compare ventilation defect percentage measurements that were obtained in 129 xenon MRI. So these were performed in stable pediatric CF subjects at these 2 institutions, and it consisted of a total of 28 participants. Eight were actually healthy and 18 with CF. And what you see by these percentage values before we even progress to the bar graphs is that you have pretty good inter-rater agreement in the ventilation defect percentage for both site 1 and 2. And when you look at these in aggregate, these actually showed that there was quite good agreement across the 2 sites and reviewers. This is important because then we can take experiences like this into broader applications. So one of the concepts that's being discussed for these types of therapies, particularly with smaller number of subjects that can be enrolled in trials, is the ability to refer subjects from one geographic area potentially to another to be able to enlist in a trial where they qualify. So being able to have multiple center consortia that can actually conduct these types of imaging and have agreement upon the standardization of how we score and interpret these is critical. So it's nice to see that in the last year there are protocols coming out for these multi-site trials to become standardized and the development of the Xenon MRI Clinical Trial Consortium. So that moves me into outcome measures that are under development for consideration. And so this is a discussion of micro-optical coherence tomography. This is actually pioneered by Dr. Steve Rowe, who's in the back of the room. I certainly can't speak to this better than he can. But this is reflecting measures of physiologic products of CFTR function, including the periciliary layer, airway surface liquid, mucociliary clearance, and the percent of active cilia at the epithelial level. This is potentially valuable for proof of concept data with CFTR modulators. Those data are under review right now for human nasal imaging, and this has also had in vitro and animal study applications as well. And this is providing fluorescence-based CFTR function information on isolated airway cells in ex vivo already, but the proof of concept and normative data have already been established in the NARS. Unfortunately, this is currently used on a single center basis at the University of Alabama, and the lung probe has been conceived but has not yet been implemented. So we would really love to see that further developed since we are talking about looking at increased numbers of studies that are looking at lung delivery. So ultimately, we need proof of concept and normative data in the lower airways to have a better grasp on how to move forward with this. But this video was shared next by Dr. K. Vijay Kumari and Dr. Steve Rowe. And so this is just a movie of the micro-optical coherence tomography in action. And you can see in blue the airway surface liquid that's moving because of the activity of the periciliary layer above the epithelial cells that are shown there. And I just clicked the wrong direction, sorry. This is taken together to show multiple images that are reflecting these measures of airway dehydration and delayed mucus transport. So that's a bit of a walk through several different categories of types of biomarkers. That's certainly not comprehensive. I co-chair the clinical working group subgroup of the genetic therapies working group with the CF Foundation with Dr. Steve Rowe. And some of the exploratory points that we've also discussed are changes in quantitative microbiology, changes in sputum rheology. So there are many other factors that could be considered, but we simply need to have more experience with this. And so I think we are still at a stage of more questions than answers when it comes to these particular types of measures in genetic therapies. So finally, as my mentor, Mike Knowles, who I think left the room, used to say, I'm just a treating kind of doctor. So when I think about outcomes in these studies, I'm so used to thinking about these clinical outcome measures that we typically think about, like FEV1, multiple breath washout, lung clearance index, patient-reported outcomes, pulmonary exacerbations, all of which are critically important. But it's also important for people in the community to recognize that these are really measures of outcomes that reflect efficacy, whereas right now we're talking very clearly about biomarkers for proof of concept. So as we move forward, there are certainly challenges ahead. Some of the key considerations to consider are the distribution of the drug delivery and how that may be affected by regional sampling and the heterogeneity that may occur of CFTR correction. We could be biopsying an area that just happened to be missed in deposition, for example. Participant tolerance of procedures, including factors to consider like sedation and cough. The availability of sites, the tools, the standardized operating procedures, and the standardization of measures themselves. So I do think that we are at a stage of more questions than answers, but hopefully that's what will make this panel discussion fun with my co-chair and Dr. J.P. Clancy. I want to thank Dr. Eric Sorscher for inviting us to do this symposium with you all today, my co-speakers and the members of the Genetic Therapies Working Group, and Jennifer Goralski. Thank you.
This is going to be cozy. So I think we're going to have all of the speakers come up, and I am sort of a pinch hitter because it would kind of look weird if a speaker was also a moderator, so that's my role here. And please feel free to offer up any questions in the chat, in online, or if you want, you can raise your hand and ask a question. That is okay, all right? It's a small enough room, I think we can do that safely and effectively to make sure we feel, like we're part of this discussion. And so I have my notes, and then I have your notes. So I'm trying to decide which ones I'm going to start with. But I think I'm going to start with one that was more recently submitted, and first of all, very complimentary to talks. But maybe while it's directed at Deepika, please, others feel free to jump in. Do we see a role for single-cell RNA-seq as an outcome measure, or a biomarker potentially of relevant to developing new genetic-based therapies?
So you would go to my favorite space first, of course.
There we go.
Thanks.
Perfect.
I think that there is potentially terrific value, especially as we continue to discover and learn more about the types of cells that are expressing and the impact that they may have in terms of correcting CFTR function. But the challenges of using these in multi-center genetic therapy trials is going to be certainly that this requires live cell use, first and foremost, and also that there's factors to consider of expense and availability at these centers to conduct this, particularly in the analysis with the bioinformatics as well. So on the pros side, there's absolutely the ability to get more sensitive and precise measures, but on the cons, it may prove challenging from a multi-center basis.
Maybe just to further, for anyone who wants to jump in, what type of work would really help make that move forward? Any thoughts about that? Experience from doing this with in other sort of laboratory-based studies, what would really be necessary for us to be able to advance this? Because I think we all see the power, but we also see the challenges.
Well, I see that there are two different limitations. I mean, there are the clinical application of just doing it correctly. Yeah. That's a challenge. But I also see that, for many of the vector systems that are used now, there are molecular challenges. For example, an LMP, having mRNA present or bound to a cell does not mean that RNA is actually being translated. If you're using AAV, the DNA genome can be in great excess of, those genomes that made it to the nucleus and are expressing mRNA. So new technologies that would differentiate between DNA and RNA in, deep sequencing, like methylation of DNA sequences and being able to decode that bioinformatically, I think would really help the field. I mean, a plasmid-mediated approach, it's near impossible to detect mRNA, even if there's expression, because the plasmid is in such great excess.
That's interesting. thank you for that input. Go ahead, Wanda.
Yeah, so I'm wondering if maybe some of these newer proteomics methods might be better to detect actual CFTR protein. The next level might actually be more specific with all of these real problems that John just mentioned.
Do you feel comfortable speaking to the sensitivity question? That's my only question that comes to my mind, but I don't consider myself a proteomic expert.
No, but I mean, I don't feel comfortable because we're just getting started in it. We're just starting to do it, and so I keep your ears and eyes open for those methods. They're becoming very spatial in their ability, like spatial proteomics methods, and yeah, CFTR might be a problem with the sensitivity, but we should look for it in these data sets and see if we can see it.
Fair enough.
I do think it would have to, it would have to be null, patients, like stop codon patients. Otherwise, you're going to have endogenous CFTR, and you won't be able to differentiate except for the site of the mutation, which, maybe down the road, detecting one peptide, with adequate sensitivity would be there, and specificity, though, would probably be a problem. Because PRM methods, need multiple peptides that are unique to that genome to quantify the protein. So,
Is it feasible to think about, maybe this is a third rail, but to think about having sort of a barcoded trans, either gene, mRNA, or even with, a peptide fragment that's not necessarily endogenous, but allows you to follow?
I think, the barcode to the transgene, that definitely makes sense. We're doing that to differentiate repeat administration of viruses in the RNA that are produced from the first infection versus the second. But changing the amino acid sequence itself for proteomics, I think, would be a risk that, would be seen as foreign and create a different immune response against the transgene.
Do we know that? No.
Do we know it for single point mutations in CFTR? If you go in with normal CFTR, are you going to create an immune response from that particular mutation? I don't think we know yet. That's a very small mutation, so.
Right.
Yeah.
I'm just being provocative, that's all. Just trying. Any other thoughts, please jump in.
Well, the single cell, Deepika brought this up, but, it's so expensive what you get one chance. You go in, you do a scrape, you get one chance. What if you miss the spot where you really got good transfection, or you capture the one where you got good transfection and you missed the 90% where you didn't? The inability to sample lots of different places to really get a picture might limit our ability to use those methods, I think.
Question out there, too.
Pardon me?
There's a question.
Oh, there's a hand up, so, yes, please, go ahead, Hillary.
I was more thinking of it potentially as a preclinical measure, and I think that would potentially make it more feasible, but there's definitely some concerns about sensitivity. So not to complicate it even more, but I did kind of want to follow up on clinical risk.
Just in case there's an online question, or listeners here may not have picked up on that. It was just questions about the potential role of single cell in the preclinical model systems and actually the development before we get to the clinic.
We haven't done it. I think it's a good question. I don't think you could do it with CFTR because it's too large. So the vectors that we have right now, to do what you're talking about, you have to be able to differentiate the DNA genome from the RNA. So you'd have to have an intron, in the AAV vector or bocavirus vector or whatever it is. And then you might be able to, from the sequencing, you're going to pick up both the DNA as well as the RNA. It's an interesting question. I mean, if you had a very strong promoter, would CFTR even rank up to the point where you'd expect to see it? Because with gene editing, I mean, now, the limits of sensitivity of detecting CFTR in various cell types is an issue. It's pretty low, and it can function at very low levels in a cell. So, it's a good question.
So I am going to ask one from here, and then I'll go back to the audience. Just as I think I'd love input from a variety of people. What's the right cell for CFTR expression, and does it matter if it's in the wrong cell? Thoughts? And maybe cells is a better word.
So we had a submucosal gland symposium yesterday, and after all the discussion of submucosal gland, I asked the panelists, are the submucosal glands a target for CFTR gene therapy? And no one would commit to it. So yeah, it's a tough question. I mean, certainly, if CFTR is being expressed in the distal airway, and that's where you see distal pathology, that has to be considered a target, right? CFTR expressing cells are the target, in my opinion.
I'm a little bit more hopeful that we don't have to hit, all the cells that express CFTR, and it's basically based on in vitro ALI cultures where you're transfecting with a vector that's not cell-specific, and you can correct, up to wild-type levels, chloride transport, and the ASL height. And it's debated, though, because there are a number in the field that have either expressed ciliated specific or not, and see complementation or don't see complementation. So it's a really important question. We just don't have the tools to ask it to, in an animal that reflects the human disease phenotype, induce CFTR in a cell that doesn't normally express it, and figure out, does it prevent disease on a CFTR knockout background? And, I hope we can do it.
Yeah, and I'll just add to that this is why we're encouraging people to look at protein expression and correlate it then to observe function. So we can look at that cellular localization and see how it affects function. And I think that that's not clearly established, and I'll go one step further. The expression of RNA is not well correlated to expression of protein, is not well correlated to function. And so that's why we're asking sponsors to be very systematic in how they do these early proof of concept experiments in HBE cells so that we can really establish that and one step further take it in vivo.
Yeah. And I'll just comment, I think the iPSC, area can really contribute to that. They're much easier to genetically manipulate in ways that you can ask a very specific question in a rigorous way and get a solid answer.
Did have a question from the audience? Go ahead.
I was curious, and research was being spoken of the stuff and especially knowing very well that international measures on function that is not a track
No, it's a good question. Actually, that's just the reason that I was kind of putting the more clinical measures that we're used to looking at together at the very end, because the focus of the discussion today for this symposium is for proof of concept studies. So I do not question at all that people would be looking at lung function measures to ensure safety in these early phase trials and to show that there was no concerning bronchoreactivity or bronchospasm or sudden decreases in FEV1. But in terms of these just simple proof of, not simple, in terms of simply looking for the detection of CFTR in proof of concept studies, I would not expect that we would be looking for changes in FEV1.
Yeah, certainly John can speak to it. I think it's safe to say that how a PFT is done in a lab, I'm sorry, in an animal model is light years different than what we do in the clinic.
Yeah, we spent a lot of work. It's more similar to, infant PFTs because, they can't expire or inspire on command. So, whether that's responsible for differences in, FEV trends or, the fact that FEV over FVC goes up in the ferret with disease progression. I don't know. I've talked to, JP's seen all that.
I have
seen the
seen
paper. So I don't know. But it's definitely a chronic disease state. I mean, the changes that occur within months of a G551D animal coming off VX770 are not significant by PFT, whereas they are by mucociliary clearance. So, when you think about an endpoint for gene therapy, you really don't want to have to wait 3/4 of a year to know.
So I had a question, and we'll go to you just after this one, had come in from one of the persons here, just around thinking about if you were going to thinking about doing immunogenicity studies to help guide some of your clinical programs, and you showed a little bit of work with this, John. I'm just kind of curious. I'd be curious both from a smaller and a larger animal model perspective. Where would the data that would be generated in a smaller animal differ significantly compared to a larger animal? Do we know?
I can't speak, for rat and rabbit, but, various inbred mouse strains have different immune responses, and they're just different. And, whether or not you could get the same type of immune response from a rabbit that was outbred, or what the strains would be that you'd have to outbred, it could be possible. But, the entire field tends to, inbreed because it's more reproducible. Well, the fact of the matter is, humans are not inbred. They're not reproducible. We see, a large amount of variation in genotype, phenotype, even with the same mutations. And certainly the immunology is going to reflect that, I would think, as well. And the ferrets reflect it.
Yeah, on the other hand, if neutralizing antibody responses can be seen in these smaller animals, just like we would expect in larger animals. So I think it depends on the granularity of your question. All the animal models, to my knowledge, have B cells and T cells and macrophages and neutrophils and the major cell types. And so, yeah, if you're just asking, is your vector immunogenic, I think you could answer that maybe in a small animal model. If you're asking what specific cell type in that animal is responsible for that, maybe that would be a little bit more difficult.
Thank you, and I appreciate that. Question, I think, from the audience? Yes, go ahead. So, question, about, I guess I'm going to paraphrase it as super expressors. Is there a problem with having a super expression of CFTR in the airway? We know that can be a problem in the GI tract. If you turn on CFTR and it's not turned off, that's a problem, cholera. But anyway, just kind of curious, is there any reason to suspect that, any data that would support that or address that from animal model studies that we know of?
Well, we know from human air-liquid interface cultures, if you use a vector, it's not specific and you can dose up. We haven't gotten to the point where, other than you're getting toxicity from the vector in the epithelium, seen a problem with overexpressing CFTR. But that's not always the case. Mike Welsh and his group had some work with adenovirus that could express really a ton of CFTR and suggested that it went basolaterally and basically shorted out the epithelium. I'm hoping that, you overexpress CFTR in a non-ionocyte and it adopts an ionocyte-like phenotype because it has so much CFTR. I don't think, at least the ALI cultures to some extent, to me, reflect that that's happening. Maybe it's a secretory cell, maybe it's not, ciliated cell, but there's so few ionocytes, and these vectors are not ionocyte specific, that the level of functional correction in the amount of hydration of the ASL and the amount of electrolyte anion transport just doesn't suggest you're correcting the ionocytes to reverse what we see when we knock them out. So,
And
I was going to say exactly what he said, that, and I'll paraphrase into the mic, that the foundation is supporting a study with Case to look at this exact question in mice. So if you overexpress CFTR, is it detrimental? Is it detrimental in specific organs or specific cells? And we're starting to answer that question because we realize it's a major gap moving forward. Mitch, did you want to add something?
CFTR is expressed much more in the villi than the peripheral CFTR normally, but yet it completely prevents obstruction. So, to John's point, it may not need to be
Perfect.
clinically good enough, may not have to be strictly in the right place. And that transgene also is expressed in a lot of other cells in the mice that don't look like the CFTR, and that doesn't seem to hurt them. So, I think there's a lot that people look out here.
that's a good point.
I'll just add one last point on this topic, and that was actually that I recall asking JP, is this going to be a problem when we were, as a therapeutic development network community, discussing the trial of a treatment that involved a CFTR amplifier to actually increase the production of CFTR before then modulator therapy acting upon it. That program was stopped, but not on the basis of safety, and certainly the preclinical data did not hazard that from being able to move forward in a trial by the FDA. So we've actually already seen some of these concepts taken into clinical trial.
So kind of an interesting question that sort of comes in from a sort of a perspective of like a DSMB member or something. So sometimes those who are looking at protocols, having things submitted from sponsors, have basically highlighted that there's really not a validated CF animal model that predicts human results. So in that context, knowing that a negative study in in an animal model may, if only, is only going to hurt you. How do we sort of navigate that sort of question? When there's academic reasons clearly to use and to study the animal models, but there may be some business risk, and I'd just be kind of curious what people's thoughts are about that consideration.
Yeah, I can start. So as somebody who looks at protocols and packages, I can say that we think of all the data as being additive. So if you have one study that's negative, I don't think that from our perspective, that's the nail in the coffin. If you have 5 studies and 5 animal models and they're all negative, that's not a good sign. So I think it really comes down to how you put that package together and building that conclusive evidence. Show us where it goes. Show us where it goes in an animal model that has CF lung physiology. Show us that the protein's expressed. Show us that you can detect function. And I think those really add up to support. And I would say that we don't take one single assay as, a make it or break it, but it's really the sum of everything together.
I'll also comment that sometimes it's not going to be feasible to test in the animal model. I mean, if you have a serotype that you feel fairly confident through other models is going to be tropic to human in the regions that it needs to transduce, and you do the experiment in ferret, and it all goes to the alveoli and alveolar type 2 cells, I mean, that testing efficacy in that model makes no sense. So in that case, you can't get around things that, the virology that's going to be species specific. Personally, I think the field needs to move in the direction where they're identifying vectors that adequately transduce both human and the models because, if we can't solve all the problems, with the first run, we have to go back and figure out how we're going to do that effectively in conjunction with the models, and then we need to have new types of tools and vectors to deliver the genes.
Yep, question, Rabin?
This is Rabin from Emory. John, I think your presentation was absolutely beautiful, and not to say the other ones were not, but I really love the other ones. With regards to efficiency of delivery and the mucus barrier and all the other things that are seen in the human, in your experience, what would you say is the amount of vector that actually goes to the epithelial cells when you have the mucus, the actin, the DNA, the neutrophils, the bacteria?
You mean, first of all, I also want to take the opportunity to, I forgot my last slide, which was acknowledgements, and I've been thinking about it since I sat down. Thank you to all my lab. So it wasn't a great presentation. So what are the barriers to, how much vector makes it to the epithelium in a diseased lung, right? Yeah, we haven't labeled the virus and looked at that. We're just now doing sort of the efficacy studies that are saying how much of the vector actually made it and transduced, not actually made it there. So I think they're important studies to do, and it's going to take different approaches to look at that. But I would like to comment, though, that, I think just to rebut a little bit of what Kara said, that, you need to get past all these obstacles. I think, this first in with the gene therapy, we're not going to solve all the problems. Getting through the mucus and all that sort of stuff. We're going to have to come up with creative ways if there are problems, like lavaging the whole lung prior to using the vector. Because, until we can begin to crawl and then walk, trying to, develop that a mucolytic that's going to be delivered simultaneously, with the viral vector is really complex. And, just getting all that through the INDs is going to be very difficult. So, I think we have to come up with reasonable endpoints that give a very firm yes or no, did this work? What was the barrier? If there's a barrier, get out of the way before you test the vector. And I'm hoping, this would be more challenging, with the TDN to try to manage this, but, the better off the patients are from a mucus standpoint and bacterial infection, I think the more definitive results we're going to get with the initial wave of the gene therapy vectors.
As a quick follow-up, just very specifically, do you see a lot of scavenging by immune cells?
It's interesting. We haven't looked at that, but the one that's possible.
Yeah, if you don't mind, thanks.
Do we see a lot of scavenging by immune cells? Well, the picture, we don't know. The picture I showed where there's a lot of green in the lumen, that could be neutrophils that are being transfected and they're just accumulated within the mucus and blocking, transfection from the epithelium, but we don't know.
I would say that in some preclinical data, we do see that.
Mm-hmm. So kind of a high-level question. Thoughts about an organized, I'm going to describe it as the TDN for animals. In other words, some sort of a organized animal model system testing program that goes beyond one animal. In other words, tying these different programs together in some way, shape, or form. Thoughts? Insanity?
I don't know about an animal TDN, but the foundation is supporting an academic program that is looking to correlate delivery of different vehicles across animal species and then in cells from those same species. So I think that will be helpful in figuring out how results from, a mouse may correlate to results in a ferret. Unfortunately, they aren't also testing in humans, so I think that clinical piece or that correlation to what will happen in humans is still missing.
Yes, question.
On the point that maybe getting CFTR to some cells that maybe normally might not express it, then maybe the follow-up question is, maybe instead of looking for a specific cell, you should be looking for the longest lived cell to actually make these gene therapy approaches more cost-effective and maybe the last 7 years, or something like that. Can we try to answer that question using the different animal models that are available to us? Like how long does a ciliated cell live in a pig versus a ferret versus a mouse? And maybe that would inform you.
I guess I remember Eric Alton yesterday saying that he's lived for a range of 3 months to 18 months, and I have no clue about the turnover of an animal relative to human. I'm looking at the anatomist over here, but any thoughts?
Well, I mean, the purest way to do it is like Bridget Hogan did it in the mouse. So you lineage trace, the ciliated cells with the cell-specific Cree, and you look at the half-life of those cells in the animal. So,
Do we know if there's a lot of variability across animal species? I doubt, I don't even know if someone's done a
I don't
comparative
know.
study. Yeah.
I don't know. There may be some other ways. I'm not as familiar with some of the single-cell sequencing, like mitochondrial mutations, and looking at lineage analysis using single-cell, deconvoluting that bioinformatically. There might be ways to figure out half-life of the cell that way.
Kara's given me the, okay. Got one more question. Anybody want to take, anyone have one in the audience? Yes, Brian.
So the question that I have is with, I think it was expressed on the plenary, sort of the robustness of the pipeline coming through. So I guess the question I have is, how do we handle, and John's comment that maybe it takes several years. So what are thoughts being given with respect to sort of administration of a new therapy to patients who've already seen an existing genetic therapy? Are there ways that that can be modeled in sort of preclinical? But I'm concerned about how do we distinguish sort of the benefit of a new therapy when you've experienced a prior.
So you want to?
Do you want to go ahead?
I was just going to answer or just raise the question for those who might have not heard it, was just how do we, acknowledging that there are a lot of potential studies coming down the pike with a relatively limited study population, how do we ensure that we're able to conduct these studies, and can animal models help us to sort of inform how we would strategize going forward?
And I don't know if I misinterpreted the question because I leaned in, and I'm certainly not speaking about animal models, but if part of the question was about some of the clinical trial strategies or and I see a nodding of the head, considerations about how we discern effect if someone has received a treatment and is moving into a different trial, I would simply say the following. My concern, and I think the focus of many, has actually been more on how one might be able to parse effects of safety in those circumstances. So because we're still talking about transient therapies or semi-permanent therapies that require redosing, the design would clearly have to be to afford a significant enough washout period and then some. But I think the bigger concern is actually there's also even an ethical concern about once you've enrolled into a trial, does that really then prohibit you from being able to participate in any other? And yet I can be sensitive to the other end of that argument of if you're developing antibodies, then how do we know exactly which this is coming from? So it's a quandary, and I don't have the answer, but I'm sure Dr. Mayer Hamlet does. So.
Last question, Ruben. Just
I have an early question for the panel. Once we start to try to measure CFTR instead of reporter genes, unless we really replace the pre-existing CFTR, aren't we not going to add to ER stress, and aren't we going to have another barrier to deal with?
So I guess are there unintended consequences at the cellular level for our attempts at genetic-based therapies, for example, high-level expression that could produce ER stress and so forth? So, thoughts?
I'd say hopefully we'll have the answer from Case in the next year.
Very soon, yeah.
If you get a mouse that's, CAG promoter level expression in all cells and it's alive, that's really
That's a
great
good
news.
sign.
And we hope to distinguish the effects of ER stress versus the transport, anion transport, that also might be mouse. That's overexpressed as G551D.
Thank you. Congratulations, a 40-minute Q&A. That was
Well done.
well done, well done.